Good morning, everyone. I am Mr. Ishitu Tasfai, your biology teacher. This is grade 12 biology, week 13. The lesson for this week is chapter 35, lesson 2, defenses against infection. Your textbook page 1014 all the way to 1019. Lesson objectives. At the end of this lesson, you will be able to describe the body's non-specific defenses against invading pathogens, describe the function of the immune system's specific defenses, and list the body's specific defenses against pathogens. Now let's turn our attention to the immune system in action, or the styles of the immune system functioning. What are the body's specific defenses against pathogens? The specific immune response has two main styles of action. These are the humoral immunity and the cell-mediated immunity. Humoral immunity, or the B-cell response. The immune response that defends against pathogens in the body fluids such as blood and lymph is called humoral immunity, or the B-cell response. The B-cell, as you can see on the right, is a special type of white blood cell, or B lymphocytes, that produces what is known as humoral immunity. B cells play a major role in humoral immunity by producing special type of chemicals known as antibodies. When a pathogen invades the body, its antigens are recognized by antibodies on the surface of few existing B cells. Antibodies are the main weapons of the humoral immune response. As we discussed earlier, B cells produce antibodies. As you can see on the diagram on the right, the antibodies have this type of shape. They look like the English letter Y. Okay, and they have two identical antigen binding sites. Two identical antigen binding sites here. Okay, the shapes of the binding sites enable an antibody to recognize a specific antigen with a complementary shape. When an antigen binds to antibody carried by a B cell, T cells stimulate the B cells to grow and divide rapidly to produce two main types of cells. These two main types of cells are the plasma B cells and the memory B cells. The plasma B cells, or simply called plasma cells. Plasma cells produce and release antibodies that are carried through the bloodstream. The diagram on the right shows type of plasma B cells. As you know, they produce antibodies. These are Y-shaped antibodies which are produced by these plasma cells. These antibodies recognize and bind to free-floating antigens or to antigens on the surface of pathogens. When antibodies bind to antigens, they signal other parts of the immune system to attack and destroy the invaders. Some types of antibodies can disable invaders until they are destroyed. A healthy adult can produce about 10 billion different types of antibodies, each of which can bind to a different type of antigen. This antibody diversity enables the immune system to respond to virtually any kind of other that enters the body. The second type of B cell is known as the memory B cells. Plasma cells die after an infection is gone, but some B cells that recognize a particular antigen remain alive. These cells, called memory B cells, react quickly if the same pathogen enters the body once again. A diagram here shows the structure of memory B cells. Okay, Their main job is to track, keep a record of a pathogen that once affect or infect our body, and then provide a quick reaction, which we call it secondary response, then the next time the pathogen comes around. Memory B cells rapidly produce new plasma cells to battle a returning pathogen. This secondary response occurs much faster than the first response to a pathogen. Immune memory helps provide long-term immunity to certain diseases and is the reason that vaccination works. Now, let's quickly summarize the humoral immunity diagrammatically. As you can see in this diagram, the first event that happens in humoral immunity is that antigens bind to antibodies produced by B cells. This will lead to stimulation of special type of white blood cells called helper T cells. These stimulated helper T cells activate B cells to grow and divide rapidly to produce new B cells. The activated B cells in turn form two types of B cells called plasma cells and memory B cells. These are memory B cells and these are plasma cells. The plasma cells release antibodies that capture antigens and mark them for destruction. As you can see here, the antibodies are destroying the antigens, the invaders. Then what about the plasma cells? Sorry, the memory B cells. The memory B cells respond more quickly than the B cells in the primary response, as we have said earlier, because they have a record of the pathogen in the previous exposure, and now they can produce many plasma cells, quickly, and destroy the pathogen immediately. Now to the second type of action of the immune system, called the cell-mediated immunity or the T cells response. Another part of the immune response, which depends on the action of macrophage and several types of T cells, is called the cell-mediated immunity or the T cells response. This part of the immune system defends the body against viruses, fungi, and single-celled pathogens, basically the protists. T cells also protect the body from its own cells when they become cancerous. How does the cell-mediated immunity work? As you can see on this diagram, macrophage consumes a virus or any other pathogen and displays the pathogen's antigen on its surface. Then helper T cells bind to the macrophage and are activated. The activated helper T cells will divide, and they will in turn activate B cells to produce antibodies which can kill antigens, and they do also activate cytotoxic T cells which kills infected body cells, and they also produce memory T cells which will provide the secondary response, which is very fast and immediate as compared to the T cells, the normal T cells. When a cell is infected by a pathogen or when a phagocyte consumes a pathogen, the cell displays a portion of the antigen on the outer surface of its membrane. You can see here. This membrane attachment is a signal to circulating T cells to join the complex. Activated helper T cells divide into more helper T cells which go on to activate B cells. They do also activate cytotoxic T cells and produce memory T cells. The cytotoxic T cells hunt down body cells infected with a particular antigen and kill the cells. They also kill infected cells by puncturing their membranes or initiating what is known as apoptosis, programmed cell death. On the other hand, the memory helper T cells enables the immune system to respond quickly if the same pathogen enters the body again. Another type of T cell called suppressor T cells inhibit the immune response once an infection is under control. They may also be involved in preventing autoimmune disease. Although cytotoxic T cells are helpful in the immune system, they may make the acceptance of organ transplant difficult. When an organ is transplanted from one person to another, the normal response of the recipient's immune system would be to recognize the donor's organ as non-self or any kind of invader or pathogen. T cells and proteins would damage and destroy the transplanted organ in a process known as organ rejection. To prevent organ rejection, doctors search for a donor whose cell markers are nearly identical to the cell markers of the recipient. Organ recipients must take immunosuppressant drugs, usually for the rest of their lives, to suppress the cell-mediated immune response. Now let's conclude this lesson by answering the following lesson check questions. Question number 1. List the body's non-specific defenses against pathogens. The body's non-specific defense against pathogens are the skin, tears and other secretions, versions of the inflammatory response, and interferon and fever. Describe the steps of the inflammatory response. Mast cells release histamines, which in turn stimulate blood flow. Fluid leaking from the expanded blood vessels causes swelling in the infected area. White blood cells then move into infected tissues. Many of these are phagocytes, which engulf and destroy bacteria. Question number 3. How does the immune system identify a pathogen? Well done. Antigens trigger specific immune defenses. Question number 4. How are the roles of B and T cells different, and how are their roles similar? Difference: B cells discover antigens in body fluids, while T cells are presented with antigens by infected body cells or immune system cells. Similarity: both recognize antigens. Question number 5. What are the two main styles of action of the specific immune response? The two main styles of action of the specific immune response are the humoral and cell-mediated responses. And the last question: why would a disease that destroys helper T cells also compromise the humoral response? The answer is helper T cells activate B cells. So if helper T cells are destroyed, B cells will not be activated. Thank you very much for watching and see you next time.